Welcome back to Dissect the Question. A 60-year-old man presents to his primary care physician with generalized muscle weakness and muscle wasting. His grip strength is tested and is found to be weak. Intravenous administration of which of the following compounds is most likely to increase the probability of non-selective cation channels opening at the motor end plate? So it seems like this man has some sort of myasthenia syndrome, perhaps myasthenia gravis, and weakness. His grip strength is tested. It turns out that when we administer something that's going to increase the probability of a non-selective cation channel opening at the motor end plate, we should improve neuromuscular transmission and improve the muscle weakness. And in fact, in myasthenia gravis, that's a test. We use a compound that does help open non-selective cation channels. Now, what is the non-selective cation channel we're talking about? Well, here's our motor neuron. Here are our vesicles that release acetylcholine onto a skeletal myocyte. Here's where we have acetylcholine receptors. And part of the acetylcholine receptor is the enzyme known as acetylcholinesterase, which breaks down the acetylcholine and keeps a muscle from being overstimulated. Now, if we inhibit the acetylcholinesterase, now the sodium-potassium channels, so on the motor end plate associated with these acetylcholine receptors is a channel. And that's a channel for both sodium and potassium. So it is a non-selective cation channel because both sodium and potassium are cations. This is unusual. In the brain, we don't have non-selective sodium-potassium channels. We generally have selective sodium channels and selective potassium channels. Occasionally, we may have a non-selective sodium channel, potassium channel, and calcium channel. In other words, the channel is non-selective for all three cations, sodium, potassium, and calcium. But sodium-potassium non-selective cation channels are associated with the acetylcholine receptor on the skeletal myocyte. Therefore, if we give a drug that's going to inhibit acetylcholinesterase, acetylcholine is going to increase in the synapse at the motor end plate, and we have a greater probability that the sodium-potassium non-selective cation channel that's necessary to be stimulated to get an action potential is going to be stimulated. So it looks like cholinesterase inhibitor is the best answer. However, in all multiple choice questions, we cannot ignore the others because sometimes there's two possible answers and we always have to select the best one. Let's look at the others. Botulinum toxin, what does that do? Botulinum toxin works on the vesicles of the motor end plate here. The vesicles of the motor neuron, I should say, at the motor end plate. And it blocks the release of acetylcholine. Okay? It blocks the acetylcholine. So, giving a drug that would block, that would be a botulinum toxin, it would block the release of acetylcholine. We'd have less acetylcholine, and we'd have fewer action potentials, and actually the muscle weakness would get worse. Calcium chloride and magnesium sulfate, calcium ions and magnesium ions, can both be administered intravenously to decrease excitability. Well, there's already a decrease of excitability in this person's muscle weakness, isn't there? So if there's decreased motor weakness or muscle weakness due to decreased excitability. And if we have something blocking acetylcholine receptors, for example, in myasthenia gravis, there are antibodies, MGs, there are antibodies that attack the acetylcholine receptors. So that's a state of decreased excitability. Calcium and magnesium both decrease excitability, and therefore, usually they decrease excitability of the motor neuron. So we'll put calcium here and magnesium here. When those are administered, they decrease excitability of the motor neuron. That would make the motor weakness worse. And tetrodotoxin is a blocker of specific sodium channels, not non-selective cation channels. And if we gave tetrodotoxin, it would also make the muscle weakness worse. It would not do what a cholinesterase inhibitor would do, that is to increase acetylcholine and therefore increase the probability of opening the channel to get reversal of a muscle weakness.